Thank you for tuning in. And basically, today we're going to talk about neuroendocrinology. And I can tell you up front that I am going to be giving you take-home questions. And the take-home questions come from the questions in this particular group of lectures. So as we go through, if you have any questions about the questions, I can answer them for you, because I really want you to get this information. And so here's my contact information here. And feel free to email me or call me or text me. So the neuroendocrine system obviously includes neurons, but these neurons are really special. They produce hormones and secrete it into a blood vessel. And they've become a very significant part of the endocrine system. However, the whole system also involves all of the loops and interactions with the brain from the periphery. So for example, if you have a feedback loop that affects a neurosecretory neuron, that's neuroendocrine. Anything that involves the endocrine system and the nervous system and target organ feedback. And we'll give you examples of that as we go along. So the neuroendocrine system is really more broadly defined. So let's focus on the place where it is in fact integrated, and that is the hypothalamus. You can see that right here. And I've actually colored it in black. And then the hypothalamus uses the pituitary, which is right here, the master gland. And I'll show you the parts of the pituitary. And basically the hypothalamus extends from the optic chiasm, which is right here, back to the mammillary body. So there's its front and there's its back wall. And you can see that its base here is part of the posterior pituitary, the neurohypophysis, called the median eminence. And so we'll get the boundaries in a bit. But basically, it contains groups of neurons which regulate the pituitary by sending axons to the blood supply in this region here. So I gave you lots of pictures of the hypothalamus so you can see. There it is in green. And here it is in blue in a real human brain cut in sagittal section. You can see the pituitary is missing in this section, but that's the hypothalamus there. And then if you look at the base, this is again a human brain. And here's the base of the hypothalamus, the floor. And basically there's the optic chiasm right here and here. And there are the mammillary bodies. So there's you can see it from the bottom. Now I show you this picture because I want to impress upon you the fact that all of the things I'm going to talk about, which are pretty important to our life, our quality of life, are regulated by this very small unit. So the pituitary itself, which I'm defining here, and there's connected to the hypothalamus by a stalk, it's about the size of a pea. So instantly imagine what a pea looks like, and then put a little stalk on top. And then you have a structure which is about the same size sticking up, divided by the third ventricle here. And you have this whole unit. So I guess it's like two peas right here. And what is impressive then is when you think about all the things this unit does and how it has to integrate, it's always impressed me about its size. Its size is always, small size has always impressed me. So, Motion, are you recording this?
Yes, Dr. Childs.
Oh, good. Wonderful. Wonderful.
Yeah, because you see, I already posted the PowerPoint because the students like both actually. They want to take
I think
on the
it's
PowerPoint
great.
And recording also helps them, you know.
You bet. You bet. This is much better. Yeah. Okay, so now I'll show you the divisions of the hypothalamus, which helps you identify where the different nuclei are. And the bottom line is, if you were to see a name of a nucleus, you would instantly know in many cases where it is, at least approximately where it is. So this is a frontal section, And looking straight at the hypothalamus, you see the third ventricle dividing it. So it's got two sides to it. So remember, the wall's kind of thick, and then everything, all the nuclei around the ventricle here are called periventricular. That's very cool. Or paraventricular. It doesn't matter. There's a paraventricular and there are periventricular. It's just right outside the ventricle. And then there's a group of nuclei that are in the medial section. So this is the medial column here, as I've designated down here. And then moving laterally, you have the lateral region. So if they say, well, the lateral hypothalamus or the lateral this or the lateral that, you'll know where it is in space in the hypothalamus. Now, another thing they do is that they name the nuclei after where they are with relationship to other structures. And a good example here is the optic or supraoptic. So this nucleus is just above the optic tract. And then there's a nucleus we're going to talk about in a little bit called the suprachiasmatic nucleus, and that's just above the optic chiasm. So they were very smart in naming these nuclei. Now, in naming the nuclei doesn't tell you exactly what they do, it just tells you where they are in space. So let's define, I already sort of did, a neuroendocrine neuron. And basically, it's just like any other neuron. It's got axons. There's endings of other neurons on this neuron. And it's got dendrites, and then it's got an axon, at least one, probably just one, that goes down to end on a capillary. That's the big difference, because it's going to produce a hormone and store it in secretion granules, and that's what these little green dots are. And then it's going to secrete it into the capillary. And that's the endocrine part. So it's a neuron that secretes its hormone into the bloodstream. And this is actually a cartoon of that, showing that the axons travel down, the secretion granules travel down microtubules to the ending here, and there the ending is on a capillary. And so when it's stimulated to secrete, it secretes by exocytosis, spilling its contents into the bloodstream. And because it's a neuron, it gets pulses of depolarization, influx of sodium, it gets pulses of stimuli, and itself may pulse. So it may send these secretion granules and secrete in pulses. And so some of the mechanisms behind this are not well understood, but the bottom line is it acts just like a neuron in terms of its ability to pulse and respond to action potentials. So the hypothalamic lesions, because of the importance of the hypothalamus, it can cause major problems. And this just shows you an example of one of them. If you don't control your appetite, you become obese. And these were signs that early clinicians would recognize the importance of the hypothalamus. And they discovered that lesions would affect the appetite, emotion, and they could put the lesions in different areas of the hypothalamus. And in the early days, that's how they defined where the hypothalamus was regulating. And so we're a bit more sophisticated now, but basically that was the early work. And it has very broad integrative functions. Very amazing when you think about how small it is. And so these are some of the general functions. I'm not linking them to any particular nucleus at this point, but just to show you some of these functions, which regulates appetite, blood pressure, water balance, our biological clock, and of course, all the functions of the pituitary gland, thermoregulation. And we're going to talk about a lot of these as we go through the rest of the lecture. We're not going to say much about immune functions, but most of the rest of this stuff we'll talk about. And so as you can imagine, the hypothalamus has to be, it has to integrate influences from the periphery. And it does. It can't work all by itself. And so it gets all kinds of influences. And it takes an entire course to talk about all of the inputs to the hypothalamus. And so I'm just giving you some of the main ones. Olfactory, so our smell, that would help us detect fire, for example. Visual clues about day and night are very, very important to particularly the suprachiasmatic nucleus. It receives visceral input from our GI tract and other visceral areas. And this will help us relay information about appetite. Are you full? Are you sick? Do you like that food? You know, that kind of thing. And then, of course, it receives emotion. I mean, your fear response, your stress responses, emotion from memories, your hippocampus. So we're not going to learn all, thank God, we're not going to learn all of those inputs. This would take an entire course. But just know that the hypothalamus, and this kind of shows you a general picture. You can see some of these fibers flowing in, and it'll come into the right set of neurons and help the hypothalamus do its job to respond, to integrate the response from the outside, or our perception of what's going on in the outside with what needs to be done, what our body needs to do. Now, one of the ways it integrates is around a set point. And the most easy to understand set point, to me at least, is body temperature. So we need to maintain our body temperature at 98.6. And so the hypothalamic neurons are able to receive information about, are you cold? Are you too hot? And it receives that information and it sends the right signals out. And we'll talk about that on Tuesday. And so if we are too cold, we can shiver, and basically that would produce heat, or we can sweat if we're too hot, and that will cool us off. So the hypothalamus would send signals to the appropriate neuronal systems and muscular systems and glands to allow us to respond to changes in body temperature. So this is called a homeostatic or reflexive. And the idea is to keep us at 98.6. That's the idea. And of course, the response is usually to a sensory stimulus. Now, another type of a stimulus is called positive feedback. And basically, positive feedback could be seen, for example, a really good example is with the uterus. When during childbirth, the pressure of the baby's head on the cervix will stimulate a set of nerves to go up to the hypothalamus, and those neurons will release oxytocin. Oxytocin goes into the bloodstream and starts uterine contraction. So this is a positive feedback loop. So if the baby is ready to be born, it presses on the cervix, and it's a positive feedback to the hypothalamus that says, yeah, we need more oxytocin. We need more contraction of smooth muscle in the uterus so that baby can be born. So basically, the signal from the process would be the baby pressing on the cervix. The hypothalamic neurons produce oxytocin. They'll receive that signal and they'll send oxytocin out until the baby's born. And then in the end, the process is stopped. Okay, so the other thing the hypothalamus does is regulate our rhythms. And I told you that the neurons may secrete their hormones in pulses. And anything shorter than 24 hours is called ultradian, or anything up, if you have sort of an hourly pulse, it's called corporal. We don't usually use those terms, but at least you'll have them defined. The one we use most often are the 24-hour rhythms. And that's the rhythm that's independent of a light-dark cycle. It's going to happen no matter what. And just to give you an example, when you get up in the morning, it's really important that your adrenaline, and your adrenal produce cortisol to make you alert. And basically, so your cortisol levels are highest in the morning. And that's a circadian rhythm. And then they're lowest when you need to go to sleep. And we'll show you an example of that and how it can go wrong in the case of, say, workers working at night. Now, there are other rhythms that are entrained to the light-dark cycle. Those are called diurnal. And so we'll talk about how that happens. And then there are rhythms that are secreted in seasonal patterns and seasonal breeders, for example. And so when the days are longer, you'll have breeding in order for the young to develop under warm conditions and when there's more food. So the hypothalamus does all of this. And let's talk a little bit about the regulation of the rhythms. And I think it's really important to recognize is that it all starts with our perception of the deep blue wavelength. Now, this is actual recognition of the diurnal rhythm. So the deep blue wavelength is found in white light. And when the sun rises, that form of light actually hits very specialized neurons in the retina to convert the photons to electrical signals. And these electrical signals are then sent to the suprachiasmatic nucleus. Okay, so you know where that is now. You already know that nucleus of neurons is above the optic chiasm. So this visual information is carried back to the optic chiasm, and then it goes up into the suprachiasmatic nucleus. And then the suprachiasmatic nuclei neurons are activated, and they are already maintaining a rhythm close to 24 hours. However, they use the light to reset the clock each day. So this signal goes in, and then all of the diurnal rhythms are then activated. And the suprachiasmatic nuclei neurons, and we don't have time to go into details about how this happens, but they send signals all over the body and basically reset the clock, saying it's morning, we've got to get going. And this focused signal indicates, it regulates many body functions. And so a lot of our body has its own clock, and the SCN helps integrate and regulate it. Now, one of the things that the signals do is suppress the pineal, which produces melatonin. And the pineal gland basically produces melatonin from serotonin. And this melatonin rises when it's dark, and it makes us sleepy. So you need to have it suppressed in the morning so you're not sleepy. And so the pineal gets involved in this way. It gets suppressed during the day. And then when there's no more blue wavelength, when it's dark, assuming you're not keeping night lights on or looking at blue screen, looking at screens, that kind of thing, the melatonin will rise. And so the pineal will then do its job to make us sleepy and help us get a good night's sleep. So that's kind of a general description of how these rhythms are set. And this is a picture of the suprachiasmatic nucleus. It's sitting right above the optic chiasm. Here's the third ventricle right here. And so these are other nuclei in the hypothalamus. You can see there's paraventricular nuclei. Here's some medial nuclei, lateral nuclei. And so you can see the two the two suprachiasmatic nuclei lying on just above the optic chiasm. Okay, so these are questions which you can go over, and basically, you know, they're pretty easy. True or false? The neuroendocrine system only involves the hypothalamic neurons. Nope, that's false. Basically, it involves the systems throughout the whole body, feeding back to the neuroendocrine system. So if you find neuronal cell bodies outside the third ventricle, they are in the periventricular or paraventricular. And so basically you can name the 3 regional columns. You can go back to that slide. Supraoptic suprachiasmatic is named because it lies next to the optic tract or the optic chiasm. Homeostatic or reflexive regulation is regulated around a set point. So what type of feedback do you see when there's pressure on the uterine wall or the cervix during childbirth? It's positive feedback. So these are fairly easy questions. A rhythm in a hormone that is unaffected by the light is called the circadian, and that is affected by sunrise and sunset. It's called diurnal. And which of these nuclei are critical for rhythms? That's the suprachiasmatic. So if you were to go over all of these, I could ask you a question that asked you to integrate this information, and you'd have no problem on the take-home exam. So now let's focus on the regulation of the pituitary or the hypophysis. And we're mainly going to talk about today the adenohypophysis, this region. And most of the time will be spent on the anterior lobe or the pars distalis. But embryologically, this region comes from the roof of the mouth, and it grows up to join a projection down from the hypothalamus or the brain called the neurohypophysis. And together they form the hypophysis. And we'll talk about the neurohypophysis on Tuesday. But basically, I'll give you some of the landmarks here. So the anterior lobe can be seen closest to the nose. So it'd be right here. And then right here, you see this funnel-like thing. The floor of the hypothalamus is the median eminence. And this stalk connecting to the posterior lobe is called the infundibular stalk. And then you have the posterior lobe right here. So these are some of the landmarks, and you'll be seeing them over and over again. So it won't be that difficult. And you can see how close it is to the hypothalamus. Now, the way you regulate the pituitary is through the hypothalamic nerve cell bodies. And all regulation has to go through the blood supply, which is right here in the median eminence and stalk region. And I'll show you more pictures of that as we go along. But you start with neurons up here in various nuclei, and they send their axons down to end on the blood supply in this region. And they secrete then into portal vessels that are literally carry the blood into the anterior pituitary. That's a very important concept. And I guarantee you're going to be asked to describe this concept in one of your take-home exam questions. And so then the secretion from the pituitary goes out to the venous system to the target organ. So that's your basic direction of regulation and secretion from an anterior pituitary cell. Now the target organ feeds back and tells the pituitary, stop stimulating me. And basically, so when its product gets high enough, it will feed back negatively. And many times it'll feed back negatively to the neurons. It'll also feed back negatively to the anterior pituitary cells. It's got specific targets, and it has to go through this route. It has to go all the way to its target in the anterior pituitary. And we'll be defining those targets as we go along. So the important thing is to know the route, understand how this neuroendocrine system works and how it links up. So this is just a picture of the hypothalamus. Here's the third ventricle. And then this is the median eminence. And I was telling you about it. So this is actually part of the neurohypophysis. But this happens to show a bunch of immunolabeled stores of neuropeptides. And I'll talk to you about the different types of neuropeptides in a minute. But they're stored here in the median eminence, where right on blood vessels, and where they will release their product into the blood vessels, which then will travel down to the anterior pituitary. And so there are special portal vessels that take these stores into the anterior pituitary. So here are some of the hormones we're going to talk about. We'll talk about some of these today and then the rest of them on Tuesday. But the anterior pituitary produces a number of different hormones, so it's quite diverse, and thereby regulates the various organs of the body. The posterior pituitary actually stores hormones. So its hormones are produced up in the hypothalamus. Again, we're going to be talking about that on Tuesday. And then I told you about the neuropeptides that are produced in the hypothalamus. These are all the different types that are produced, and this just kind of shows a diagram. So let's look at a group called the CRH neurons. These guys are going to stimulate a cell in the pituitary that produces ACTH. Here you go, ACTH, and they produce corticotropin releasing hormone, or CRH. So there they are in the paraventricular nucleus. And you can sit there and go, oh, well, I know where that is. It's right next to the third ventricle, and you'd be right. And so basically the CRH then is stored down here in the median eminence. And then when we have stress, the CRH is released and it's sent to the anterior pituitary. And in the anterior pituitary, you'll find the ACTH secreting cells. We'll talk about them in a minute. And they'll stimulate the adrenal to release cortisol. And that's a very rapid way. This is the way we respond to stress. And so each of these hormones, TSH, gonadotropins, growth hormone, have their own special releasing hormone that are found in different regions of the hypothalamus. And so for the next few slides, maybe all the way to the end of this lecture, we're going to go through each system, and you can understand then how it all works. So we were talking about stress. So let's start with the corticotropes. And stress is something we may perceive. It's probably a real thing, or it may be something that we're fear of. Some people afraid of flying. No matter what, it activates this particular set of neurons and pituitary cells. And so there it starts out, as I said before, with the hypothalamus and the CRH neurons, and they're sending their hormones down to the blood supply in the median eminence. And then there are portal vessels that are sending the hormone to the pituitary corticotropes. And they have a CRH receptor, so they can receive the CRH, and that stimulates them to release ACTH. And this happens in a matter of minutes, even seconds, it starts going out. So you have this very rapid rise in ACTH after a stressful situation, or you see something. It's part of the flight or fight reaction. So this rise increases ACTH in the serum, and that goes to the adrenal cortex. Now, we don't have time to go all the way to talk about the whole the endocrine part of this, but just know that the adrenal cortex has a special group of cells that produces cortisol, and cortisol rises. There's cortisol. This is what it looks like. It's a steroid. And it rises, and basically it forms part of our stress response. So here's another picture of the whole system. There are your CRH neurons up in the paraventricular nucleus. Here's the axons going down to the blood vessels. Again, use these slides to understand the circuitry. So then when I ask you a question about describe the circuitry that gets involved in response to stress, you'll be able to write this accurately. So here's these the portal vessels sending the CRH to the anterior pituitary, stimulating corticotropes to release ACTH, which then goes to the adrenal cortex. Now, what this slide is showing is feedback. And the feedback is going all the way up to the CRH neurons. So we would go crazy if cortisol didn't feedback negatively. I mean, we've got to get this thing under control after a while. And so basically it shuts down the system, helps us to calm down a bit. The only thing I don't like about this cartoon is that they've got the feedback going directly to these cells. Actually, they should go through the portal system. That's the only route here to these ACTH cells, is through this portal system. And these are just pictures showing the paraventricular nucleus. And this shows the same nucleus where you've actually localized glucocorticoids with a tracer. So you know that it's bound to these these particular this is the white here, to these particular neurons. So here you see an actual cartoon of the blood supply. And I hope I'm showing this because I think it'll help you understand it. So here's stress. Here are some CRH neurons sending their axons down. There's the median eminence stalk region, secreting CRH into these capillaries. And then these are the portal vessels that are actually sending the CRH down to the anterior pituitary. And they're stimulating the release of ACTH. So that's how the adrenal cortisol producing cells are stimulated. This just shows you a picture of an anterior lobe corticotrope in a human. And you can see that it's ready to go. It's got a lot of secretion granules that are loaded with ACTH. So, so you can see another process of one over here. These are immunolabeled for adrenocorticotropin. These are some of the other pituitary cell types. You can see some of them have tiny granules like thyroid hormone cells, thyroid stimulating hormone. Somatotropes have granules that look just like corticotrope granules, only you know they contain growth hormone, not ACTH. Prolactin cells will have very funny shaped granules. So that's how you can tell what they are. So corticotropes produce the ACTH, but it produces it as a prohormone. And you'll probably hear about the pro-opiomelanocortin. And that prohormone can actually be a prohormone for several other hormones. And of course, adrenocorticotropin is one of them. Beta endorphin is another one. And alpha melanocyte stimulating hormone is a third. And so we'll have beta endorphin released by stress to help us relax. It's also kind of like an analgesic. So it's kind of a multiplex system that sends these hormones out in cases of stress. And then this just shows you the feedback loop. I won't go over the details, but this is the feedback loop to those particular cells. Now, the CRH, ACTH, cortisol axis is basically rhythmic. And as I alluded to earlier, it is circadian. It has some diurnal influences, but basically it's circadian. So here, for example, is you can almost guess, this is night. This is when cortisol is low. And then we're awakening, here we go. And the cortisol is high and continues to be high throughout the day. That keeps us going, keeps us alert. And then it starts to go down, and here again, it's going down. So that's the rhythm, that's the circadian rhythm. But what is interesting, what happens to somebody who is a shift worker? So here's a normal sleep time, and you see the cortisol rhythms are low, and then they're high during the day, right here. But what happens if the person is working at night and sleeping during the day? Well, sadly, because this is a circadian rhythm, it's not dependent on light dark. It's the cortisol levels are still going to be high, and it really makes it difficult to sleep. And so if you know about shift workers, you know they have a hard time sleeping during the daytime because their cortisol levels are actually high. Their suprachiasmatic nucleus and their adrenal and their corticotropes. I mean, they're ignoring the day. They're ignoring everything. They're just saying, well, it's morning. We've got to get going. And so this is the normal pattern. And basically, this actually causes even more stress, and it looks like it's a bit more stressful there with even higher levels of cortisol, where the poor person is trying to sleep. So some quizzes. So why is the blood supply to the anterior pituitary efficient? Well, you know what? I didn't really tell you that. It's because you really don't need a lot in order to stimulate the anterior pituitary hormones. You just need a little bit. And in fact, they can't even measure the levels of those peptides in the portal system. And so it's very efficient and it's very rapid. And remember, it's very small. You've got like 2 peas. That's the size, connected by a tiny stalk. And so it's basically very small. Describe the circuit of hormones. You can do that now. Think about CRH, think about the circuit, think about ACTH and the adrenal. Is the rhythm secretion of ACTH and cortisol diurnal? Nope, it isn't. It's circadian. And what hormones do they produce? Well, there's beta endorphin, there's the pro-opiomelanocortin, alpha MSH, and ACTH. And then the function of ACTH is to stimulate the adrenal to produce cortisol. And then, of course, cortisol will feed back negatively onto the CRH neurons and the ACTH cells and stop it from secreting. Now let's move to growth hormone. Now, growth hormone is basically important in regulation of growth and development, but it also optimizes body composition after we've grown. So it's important throughout our entire life. And it is stimulated up in the hypothalamus by a group of neurons in the arcuate nucleus. These are the GHRH, growth hormone releasing hormone neurons. And it has another set of neurons in the periventricular nucleus called somatostatin. And these guys inhibit growth hormone. So it's got 2 groups of neurons impinging on it or regulating it. And both groups send axons down to the stalk median eminence here, and they secrete into the portal system, which carries the hormones down to bind to anterior pituitary somatotropes. These are cells that produce growth hormone. And these cells have receptors for GHRH and somatostatin. And if they see GHRH, they will release GHRH. They'll release GH. And GH then goes to the liver and releases IGF-1, insulin-like growth factor 1, which stimulates growth of bone and muscle. And so basically, the circuit is a little more complicated, but basically this is how we get growth in long bones and thickening of bones. This is how we build muscle. So this just is another picture of there. Here's the arcuate nucleus. And this also, somatostatin can come from another region of the hypothalamus as well. So it can come from a periventricular and the anterior hypothalamus. And so the bottom line is you have these 2 sets of neurons sending their hormones down to this region, the portal capillaries. In this case, they're going to regulate growth hormone cells, which is a separate group of cells. And the growth hormone then stimulates growth in long bones, stimulates the liver. Of course, all this is done through IGF-1 and strengthens muscle. Another thing growth hormone does is breaks down fat, and it goes to fat cells directly, and it breaks down fat. And so growth hormone is considered, especially as we're growing up and as we're growing older, important for optimizing body composition. And this is a picture of GHRH labeling in the arcuate nucleus. The arcuate is kind of funny because it's not really named for a region. It's just kind of over under the base of the hypothalamus here. You can see the third ventricle. And so it's really not called paraventricular. It's just called arcuate. So basically, again, we've gone through this. Growth hormone releasing hormone is released to stimulate growth hormone. And then the IGF-1 will feed back onto these neurons to stop the release of growth hormone. They'll also feed back through this system onto the pituitary growth hormone cells. So that's how your negative feedback happens. But you also have somatostatin. And somatostatin is kind of like a brake, and it's like a brake on growth hormone releasing hormone. And it's produced by the neurons. And when somatostatin is elevated, it stops growth hormone releasing hormone from stimulating growth hormone. So I'll show you a slide for the circuitry in a minute. But this is basically what somatostatin looks like. So here, for example, is a kind of the yin yang of the somatostatin. So as long as somatostatin is low, you will have high growth hormone releasing hormone, which is the dotted line here, and that stimulates high growth hormone, which is the solid line. And as somatostatin starts to rise, it inhibits growth hormone releasing hormone, which causes inhibition of growth hormone. So you can see how this thing pulses. And this looks nice and even and everything, but actually it's not super even. If you were to go and look at the pulses here, you can see it, they're very irregular. And in men have fewer pulses than women. And so you can see women, these are just example of GH pulses. And all of that is set up by somatostatin and GHRH from the hypothalamus. Okay, now what about the rhythms, the daily rhythms of growth hormone? And this is kind of where it gets interesting. Growth hormone has pulses in response to GHRH, but their highest activity is at night. So let's say you go to sleep, and I don't know if you've had a sleep lecture yet, so you know the different phases of sleep. But during slow wave sleep, you have the highest activity of growth hormone, which is kind of cool because that's the time when you can build bone and muscle, when you're a growing child. I mean, it just shows you how important slow wave sleep is. And they don't know exactly why slow wave sleep allows for this, but it's really important. So it is diurnal as well. But the most important is that you've got to get into your slow wave deep sleep, and basically you'll get growth hormone release higher. If a child doesn't go to bed on time and basically doesn't get enough sleep, this can have some bad repercussions. Also in adults, the growth may not be normal. Puberty may be a problem. There may be delay in puberty because puberty is dependent on testosterone and other gonadal hormones. Basically, all of that is connected to sleep and the right amount of growth hormone. And then I told you that growth hormone is lipolytic. So as it's building muscle, it's building bone, it's breaking down fat. So if you get a lot of exercise, you can stimulate growth hormone. Here it shows you. You can stimulate your growth hormone secretion during the day by exercising and by getting enough sleep. And collectively, you can use growth hormone to break down fat. So basically, both sleep and exercise are important in combating obesity. Now, before the epiphyseal plate, and you probably maybe don't know what the epiphyseal plate is, but this is a plate that is in your bones before puberty, and it's cartilage, and growth hormone, or IGF-1, stimulates this epiphyseal plate to grow, and that's how you have growth in long bones. And so before closure, which happens at puberty, at too much growth hormone, like in a tumor, can cause gigantism. And this is an example of a giant. Unfortunately, when you're this tall, your body cannot defend itself. And so he ended up dying of cellulitis of the feet. And so he had a pituitary growth hormone secreting tumor. I think he ended up being over 8 feet. And this just shows you the variety of growth hormone deficiency. Here, this is a giant, and this is a dwarf. So you can have a deficiency in growth hormone or growth hormone receptor and have a condition known as dwarfism. Now, if you get a growth hormone tumor after puberty, you have a condition called acromegaly. And what happens there is growth hormone will cause the thickening of bones in your jaw and extremities. And here you can see it can be quite deforming and painful. And so this is a gentleman who got, it's called acromegaly, and he had a tumor after puberty. And this shows how devastating it can be. Here's a child who became a young woman, and you can start to see some of this thickening here. Basically, it got thicker and thicker and thicker. Her bones got thicker, and it became quite deforming. And it's really sad. Endocrinologists would like to catch this long before it really deforms the face and the hands. It's really sad that they were unable to catch this disease sooner. So if I were to ask you to describe the hormones involved in the hypothalamic growth hormone IGF-1 axis, you talk about GHRH, you talk about somatostatin, growth hormone IGF-1, it's negative feedback. Before puberty, it causes growth in long bones through IGF-1. After puberty, it may cause thickening of long bones, but it also, growth hormone, is important in breaking down fat and building muscle. The rhythm of GH secretion, remember, it's important to get a good night's sleep because its strongest rhythms are found in slow wave sleep. And so this is really important for building strong bones and muscles at night. And what about a tumor producing too much growth hormone? Before puberty, you're going to have a giant, and after puberty, you'll have a condition called acromegaly. So now let's move into the last section of this talk. And we've got time. And here we're going to talk about gonadotropes and lactotropes. And these are sort of the reproductive cells. And let's start with gonadotropes. And they are stimulated by gonadotropin releasing hormone neurons. And these neurons, they send their GnRH in pulses, and they're found all along the hypothalamus, in the anterior hypothalamus, and also in the arcuate nucleus. They send their hormones to the blood vessels, saying this over and over again, which are transported down to the gonadotropes. Now, we don't have time for a detailed description of the reproductive biology of this, but basically there are 2 gonadotropins, 2 hormones that they produce. And luteinizing hormone, or LH, stimulates ovulation in the ovary. That's about as much as I'm going to say. And then in the testis, it stimulates testosterone production. Follicle stimulating hormone stimulates the growth of the ovarian follicles, which contain the oocyte. So it stimulates the development of the oocyte and the follicles that support it. In the testis, follicle stimulating hormone is important for stimulating the cells that support the sperm. So these are factors that you need to know the difference, just maybe a 1 or 2 sentence definition of these particular hormones. And they are regulated by gonadotropin releasing hormone. So again, this shows you all the different places where GnRH neurons are found along the hypothalamus, but they all end up sending their axons to the median eminence to empty into these vessels and then be transported to the gonadotropes. Now, the interesting thing is that the neurons themselves arise embryologically from the olfactory placode. I mean, it's very weird. They literally crawl out of the olfactory system and migrate to the hypothalamus, and they're distributed along these regions. So this whole thing is totally dependent on the normal development of olfaction and the olfactory system. So it's very weird. So very often, one of the things they can do is when a young man goes through puberty and or fails to go through puberty, let's say, and he notices that he doesn't have a really good sense of smell, they'll often test him and say, aha, there may be a problem here in the migratory route. So basically, you'll have the condition called positive feedback. And the positive feedback comes from the ovary here. The target organ here is ovary. And estrogen is secreted by the growing follicles. So, and of course, the follicles are stimulated by follicle stimulating hormones. And estrogen from these follicles rises and also stimulates GnRH up here. And the GnRH starts to pulse and stimulate the anterior pituitary gonadotropes. So the bottom line is the estrogen rise early in the cycle gets this going. And once basically the gonadotropes are fully mature and the pulses of GnRH are fast enough, this causes the secretion of luteinizing hormone. And basically the follicles are telling the pituitary, okay, we're getting mature and we're going to be mature enough for ovulation about mid-cycle. And it may take about 15 days, something like that, for us to get mature enough. But the estrogen is the signal for that. And then once they are mature enough, the LH will come out in a big surge. It increases to a surge. And this surge then stimulates ovulation of the mature follicles. So let's again start with the GnRH neurons are up in the hypothalamus, and they've developed GnRH neurons that can be seen by green fluorescent protein. And this is driven by the GnRH promoter. So you can study these neurons and you can actually follow them, watch them develop embryologically and follow them to their resting place, and then also stimulate them and watch their action potentials and things like that. And the interesting thing I alluded to is that when pulses are rapid, like 1 pulse per hour, LH is favored. LH secretion is favored, and FSH, that doesn't want to secrete much. But then as the GnRH pulses drop a bit, then FSH is favored. And of course, the slower pulses of GnRH are found early in the cycle. And this is when you need FSH to be stimulating the follicles. And then as the follicles are stimulating, estrogen rises, and the rise in estrogen stimulates faster pulses of GnRH. And you can see what happens. The LH will go up, and eventually you'll get an LH surge. And then, like I said, the FSH secretion is not interested in rapid pulses of GnRH, so it's just kind of poops along like this. So basically, it can favor one versus the other, even from the same groups of cells, even from the gonadotropes. Now, we don't have time to go through the whole cycle and everything that goes on, but I'm just going to show you, this shows you the LH surge in the mid-cycle. This is in the human, and there's a rise in FSH at that point, but it shows you the early rise in FSH here. And then, of course, you have the rise in estrogen, which is driving this. So it shows you all the things that are going on leading up to ovulation. Now, again, this is not a reproductive biology course. So after ovulation, you have a condition where the pituitary is shut down because the uterus is really waiting for a pregnancy. And so that's done by negative feedback. And the negative feedback comes from progesterone, and then also to a certain extent, estrogen. So estrogen has both positive and negative feedback effects. And this is the same thing in the rat or the mouse. Here's the rise in estrogen. Here's the LH surge. And the only difference is you have the estrous cycle. And so if you study reproductive biology, we'll be giving a neuroendocrine course where we'll be talking about this next fall. But the bottom line is you'll learn about the different stages of the rodent cycle and what's going on at each of these stages. But it's very similar to the human in terms of the changes in the different hormone levels. So there's a lot of negative feedback on GnRH and gonadotropes here. And then early in the cycle, before the surge, there's positive feedback. And this just shows a picture of the migration. Here you have the olfactory epithelium, and the GnRH neurons are following the tract as it's developing, the olfactory tract. So they're literally crawling along it. So this is why if there's a problem, let's say the olfactory tract doesn't develop, these neurons, and these are the little dots, these neurons cannot migrate, they can't track, and they'll literally get stuck here. They'll get stuck in this region, and they cannot regulate the pituitary from this region. So the individual has probably a perfectly well-developed pituitary, but he or she cannot respond because the GnRH neurons are in the wrong place. So if they get to migrate where they want to go, and you can see them, this is the route of migration, basically, then they'll be able to send their axons down and stimulate the pituitary. And so, what you do about these people, they don't have a good sense of smell, and they're not going to go through puberty because their GnRH neurons can't get to the pituitary. You just give them GnRH, or you can give them reproductive hormones, and they will develop because everything else is normal. It's just waiting for the GnRH neurons. And I do not know. Okay, so this is actually some pictures of migrating GnRH neurons. I thought I had this as a movie, but I don't. But there are movies out there, so you can actually see them migrating along through this system. Okay, so the last cells we're going to talk about are the prolactin cells, or the ones today. Now, the prolactin cells are really interesting because one of their major functions is that they regulate lactation. And the majority of the population don't need to be lactating. You only need to lactate after you've had a baby, and men don't need to lactate at all. And so the lactotropes are chronically inhibited, and they're inhibited by a group of neurons that produce dopamine. And these neurons are up in the arcuate nucleus. So this is the second group of inhibitory neurons I've told you about. The first one was the somatostatin neurons. But these dopamine neurons, again, they're just like others. They send their dopamine in granules down to the stalk median eminence, which is transported to the pituitary lactotropes, and basically chronically inhibit them. Now, the lactotropes are important in the development of mammary glands, and they regulate the actual production of milk, and they're stimulated by estrogen. And of course, estrogen rises in pregnancy. And so this whole system is set up so that estrogen will stimulate the development of these prolactin cells as a woman is pregnant, so that by the time she gives birth, she can produce milk as well. And so these mammary glands have a prolactin receptor and can produce milk, basically. You can develop prolactin secreting tumors, and these tumors can produce prolactin, and there'd be inappropriate lactation if a person had a prolactin tumor. So for some reason, a hormone called thyrotropin releasing hormone, TRH, also stimulates prolactin. We can't figure out why it does that, but I do want to put it in here. And we'll be talking about TRH on Tuesday when we talk about our responses to cold. And so here are some TRH neurons coming down, and it will stimulate prolactin. But we really don't know the normal reason for that. It does. And then the dopamine, of course, is inhibitory to prolactin. And so basically, again, here's the route, the portal vessels to the anterior pituitary cells. And then prolactin is secreted and stimulates the mammary gland. So now, what do you think might happen if you were to lesion the hypothalamus, a lesion right up in here, so you don't have dopamine? And workers have done that. Or if they were to take the pituitary out and grow it without any dopamine around, what will happen is the lactotropes start growing wild, literally. They will, without dopamine, they will go crazy, and they'll proliferate and they'll produce prolactin. And so clearly the chronic inhibition by dopamine is really important. And so you don't want a prolactinoma, pituitary tumor of prolactin cells. So basically, test yourself. What hypothalamic hormone stimulates gonadotropes? That's easy, gonadotropin releasing hormone. Where are they located? Well, in various places all along the anterior hypothalamus and even up to the arcuate nucleus. What are the functions of LH and FSH? Well, LH stimulates testosterone production in the testis and ovulation in the ovary. FSH stimulates follicles in the ovary, and it helps the production of spermatogenesis. Basically, it helps Sertoli cells support sperm. So basically, they're both very, very important, and they complement one another. Describe the embryologic origin of the GnRH neurons. Well, for some reason, they come from the olfactory placode. And why is a failure in the development associated with lack of smell? Well, their function and placement are totally dependent on the migration of the olfactory neurons. And these olfactory neurons need to migrate and develop so the olfactory system can develop. And the GnRH neurons just move along the fiber tracks until they get to where they want to go. And so if you don't have your olfactory neurons moving along, you have a problem. And you also have a problem with reproduction. And of course, finally, we just talked about this. Lactotropes are the only cell type that's chronically inhibited. And what do you think might happen if there's a lesion? Well, they will grow wild. There will be too many lactotropes. They'll proliferate and secrete too much prolactin. So I think we've got time for some questions. If anybody has any questions, I'll stop sharing my screen. And hopefully this was clear. Any questions? I didn't think to just recommend that you write questions out too, because I'm happy if you ever want to do that, you can. But feel free to send me questions by my email if you want to. And on Tuesday, we will look at the regulation of the thyroid. We'll look at the pars nervosa and its regulation. And then we will look at regulation of appetite. So that'll be fun. Okay.
So going back to the
Sure.
homeostatic responses, would
Sure.
weight be considered a set point as well?
Would what?
Would weight,
what?
Does weight have a set point?
Oh, that is such a good question. Oh my gosh. Yes. Yes. I would say so. And I don't know about you, but I'm going through the Noom program, which is a really good intellectually driven weight loss program. And they talk about different set points and happy points and things like that. And yeah, there's a general belief that there is a set point. And but there's so many factors that go into it, and including stress. Cortisol makes you want to eat, for example, and basically your exercise, and there's all kinds of things that go into it. But yeah, there's a set point. So I don't know if it's happened to you, but if you ever try to lose weight, there's times of where you're just plateauing, and they think that is a set point. Your body is defending itself. And what is true, and we'll talk about this when we talk about appetite regulation, as you lose weight, your metabolism slows. Your body goes into defense mode and immediately says, oh my gosh, we got a slow metabolism, we're losing weight. We don't, this can't happen. And so that also contributes to the set point. So, and a lot of that is done through the hypothalamus. So yeah, very long answer.
Dr. Childs, I had a quick pituitary question for you.
Sure.
So when you are looking at a pituitary, I'm specifically asking about mice just because of like the nature of what we do.
Sure.
Is there a way that you can visually tell the difference between the anterior and the posterior pituitary, or is that just by cell type and you would have to?
So I sort of, as I was developing my expertise in the pituitary, I used rats a lot. And so it's really easy in the rat. What happens is if you look down, if you open, you know how you open up the skull and you flip the brain back and you look down on it, it looks like there's a button. Mm-hmm. Do you see? It's kind of like a little white spot on top of it's like a button, and it almost looks like a kind of like a little ruffles around the button. I don't know if you can picture that, but there's a little button right on top. That button right in the center, and you have the two sort of lateral lobes on each side. Mm-hmm. That button is the posterior lobe.
Oh, okay, interesting.
So I'll try to find a picture of it and show it to you. In fact, that'd be really good. And so that button on top, and so in the rat, we used to routinely take a scapula and just kind of scoop that, because there's a separation between the intermediate lobe and the anterior lobe. Mm-hmm. And so we'd pull away the posterior lobe, and the intermediate lobe would come away too, because it's attached to the posterior lobe. So then we'd have just anterior lobe cells. Mm-hmm. But I don't think we do that with the mouse. The mouse is so tiny. Mm-hmm. And I know my eyesight went beyond, but it's so tiny. And most of the people in the lab can do it really well, but I've always had a hard time with the mouse pituitary. You've probably learned how to do it, right?
Yes, yeah.
You can get it, but just look to see. And there is a way. If you take a very thin kind of a stick or probe motion, you've done it.
Yes. But yes, as Dr. Childs, as you said, in rat it's so much easier, but in mice.
Yeah, the rat is really easy. But if you take a little probe and you can lift it up, and you can actually pull away the posterior lobe, but at least you can see it. And so in all of our preparations, we have posterior lobe attached. We just leave it attached. So
thank you. I've been curious about that when I've been harvesting pituitaries.
Yeah. You just make sure it's all intact as you look down on it, and or you maybe use a magnifying glass. You should see it right in the center, and then the two lateral lobes will kind of go up, sort of, And then it's right. It's like a little thing that sticks up.
Okay, thank you.
So, yeah. So, any other questions? Okay, sounds good. So same time on Tuesday, right, everybody? We're good. Okay. Thanks a lot.
Thank
Have
you.
a good weekend.
Thank you.